Hello, my name is Rayan Morad. Today we're going to be solving the Bioinformatics project. We're going to copy the sequence and we're going to go into these tabs are open to save time later. So we're going to go here into BLAST and we're going to open nucleotide BLAST. We're going to paste the sequence and BLAST. While it loads, let's read the rest of the questions. Here we have most close host organism, name, genus, species, strain, and determine the number of SNPs present in the sequence. Alright, in order to do these, we need for the class to complete. Okay, we're gonna open alignment, and here we're gonna find the wild type over here. This is the wild type gene sequence. This is the name and genus and all of those of the organism, and the SNPs, we're gonna minus this number from this number, which is gonna give us 9, so we have 9 SNPs. So those are the first 3 questions. We're going to skip to 9 and 10 in order to save time. We're going to open the organism's genome, which we just got. And we're gonna take this nucleotide sequence, the nucleotide sequence that we got over here, which is the wild type, and copy it. And then we're gonna go into Eugene. Here we're gonna go into find pattern, and we're gonna paste this sequence, and we're gonna let it run. While it runs, we're going to go into here, and we're going to take this sequence and paste it here, and then we're going to write new temp, and we're going to pick the saving location, which is downloads, and we're going to create. Yes. Okay. It was already I was practicing already, so here we're going to go into analyze, find pattern, and JET. We found the pattern. Here it is. Then we're going to go into actions, analyze, find the tandem repeats, and we're going to choose whole sequence. Here they are. Gave us 2 because we did 2 times. Then here we have design primer inclusive of the start and stop codon, and visualize the position on the gene using your selected software. Okay, here we're gonna go into actions, analyze, and we're going to go into primer 3 design. Let's close this, and we're going to go into we're gonna make sure everything is okay. Then we're gonna type pick primer. Our primers are already ready here. So we're going to choose primers that are on the stop and the start codon. So here we have the start codon of this ORF sequence. It's on it, and here we have the stop codon of this ORF sequence, and we have this primer on it. So we're going to add the PAM-H1 to both of those primers. So here we did that. We added, we enabled the editing mode from here, switch on editing mode, and we type down the nucleotide sequence of PAM-H1, which is GGATC, and we went to this primer as well, and we did the same. Then we went, the next part we need to do is clone a genetically modified gene into Pac19 plasmid. Okay, in order to save time, we already have Pac19 downloaded from NCBI. Both of those are downloaded from NCBI. Open. Here it is. We're gonna go into settings, analyze, and we're going to cloning digest into fragments, PAM-H, cut, and we're gonna go into the mutant. We're going into the mutated gene, and we're going to digest into fragments, PAM and PAM H1 and cut. Then we're gonna go into cloning again and create fragment. Nope, sorry, create molecule. We're going to choose our fragments. So we're gonna add this and this. It's because there are two I think we added PAM H 2 times, but it's fine. We're gonna choose these two, then we're going to choose, oh, it's because we already did this before, sorry. We're gonna add these two. Here we go. Then okay. And we have the new molecule, which is circular, and so we can make it circular, and here it is. Let's go back to step 9 because it's done. Here we found our sequence. It's at 5,000 this number. So we're going to go back. We're gonna go upstream, which is to the left over here, and we're going. Let's turn on the ORF. Okay. We're gonna go back from this sequence 10,000 k PS pairs. So let's which is gonna be this number. We're gonna go back to 5,000 518 735. Go, and we're gonna find this sequence. Here we're gonna find this one, which is 1884, which is close to the number we're looking for, and it's in this location. So now 9 is completed, and for 10 we need to find an ORF sequence within those 1000 sequence. So we're gonna go into here, and we're gonna press here again, and we're gonna get this sequence, which is 170. So that's 9 and 10 completed. Now we need to generate a protein sequence for the mutated DNA sequence utilizing AlphaFold. So we're gonna go back to our mutant gene, and we're gonna pick this ORF. If we press copy and paste, we're gonna find the copy annotated amino acid sequence. Now we're gonna go into here, and we're gonna go into AlphaFold, which we already had opened since it takes a long time to load, but we're gonna paste our sequence over here. Then go down, and here we have all of our generated data, and it's at the end it saves a zip file. So we're gonna go into let's go to the next step. The next question, we finished this question. Now conduct a comprehensive structural analysis between the predicted 3D structure and the experimental determined 3D structure of the original wild type gene. Okay, for the original wild type gene, we need to take the mutated sequence, copy, and we're gonna go into here. We're gonna go into bimodal protein, and we're gonna paste our sequence. Copy, paste. Then we're gonna transcribe and translate, and now that we have this amino acid sequence, we're gonna copy, go into here, go to blast, protein blast, and blast this protein sequence. While this loads, we're gonna open Bimol. Let's close. Let's just hold on. Let's close this. We don't need this anymore. Let's close it. Let's open Bimol, and let's select file. Hold on. Let's open our files. Here we have the test. we're going to unzip, extract files. Okay, and we have a bunch of structures. We're just gonna pick one of them, since we can only align between the mutated and the one and the wild type. So let's open, and let's pick from the downloads. Let's go to test, and let's pick this one, or this one, the first one. Here's the structure. Now let's go back into the blast is complete, so we're gonna get the name of the protein. So we're just gonna take the whole, and we're gonna go the whole name and go to NCBI. Okay, that is wrong. NCBI. And we're gonna go into structure, paste, go. Let's remove the brackets. The brackets must be removed. And we're gonna go into this structure, and we're gonna download it. Once it's downloaded, let's go back to Biomol and open. We're gonna go to downloads again, and we're gonna go back to downloads, and here. Now we need to align them, so we're gonna go into here and type align. Let's move this. 1A 6x. Then let's go into here and copy this name over here, which is our wild type, our mutated, and paste, and don't forget to add space. We didn't paste it. Hold on. Copy. Okay. Now if we press enter, we get an RMS value, and now that they're aligned, let's finish the last two steps. Let's open KEGG. Alright, for this we're gonna need the name. We need to get the pyruvate metabolism pathway from KEGG. KEGG. And let's open string. Okay, we're gonna go on KEGG. We're going to paste our protein. Then we're gonna minus all that. Start. Go into here, and we're gonna open the pyruvate metabolism pathway. And while that loads, let's just press this. Escherichia coli. Let's just remove this. Escherichia coli. Search. And here we have it. We're going to continue, and let's go back here real quickly. This is the pyruvate pathway. Here's our enzyme, and we're gonna go down here for the last question. We need to rank 3 genes based on their proximity. So let's go into string. Here are 3 genes. ACCD is the closest to our protein. ACC is midway, and FABH is the farthest, has the farthest proximity to our protein. And that's the bioinformatics assignment. Thank you for listening.